Welcome to the Europa Data Explorer single-cell RNA-Seq analysis. These data come from a study on COVID-19 and flu. You can download them directly in Europa using our geo integration. First, let's go to our Studies tab. We see our single-cell study named by the geo accession ID. We open the study on the right using the Open Study icon. In the upper left, we see our data segments. Each of these segments represents a single sample. Select a sample to see its report in the lower right. Each report includes standard quality plots such as genes by counts, total counts, and percent mitochondrial. Note that to start, we have no filtering parameters set. Using the Rerun QC icon, we can set quality filters for genes and cells. These are run on each of the samples selected in the top section. Here, let's input the same settings that were used in the original publication, including 200 to 10,000 genes, a maximum of 15% mitochondrial, expression in at least one cell, and doublet removal. Once complete, we see that the single-cell report has been updated with our parameters and pass filter cells and genes. Note that the dataset segments in the upper left still display the total raw cells and genes for comparison. We can rerun QC as many times as we'd like, including running on subsets of samples with different parameters. Once we're happy with QC, we select the Create Merged Dataset icon to combine our samples together. At this stage, we can deselect any sample that fails QC and not include it in analysis. Here we keep all samples. Now let's minimize the segment section since we're complete with QC. By default, the merged dataset undergoes standard normalization, PCA, and UMAP. Before viewing that, let's add sample annotations. In the upper right, we see our samples represented as test tubes. Click the Manage Annotations icon. In the Annotations Manager, we can add annotations directly or upload a file. To get matching names, let's export the current annotations and edit them locally. Here, I've pre-made an annotations table for this demo. We add it to the EuropaXp file and make sure all of the sample names match. Then we take the same file and import it back into the Annotation Manager. We select which columns to use to match sample names and note if there are any mismatches. Then we select the annotations to add. Here, we'll add everything. Finally, we can make further edits to the annotations we just added. Europa automatically detects group versus numeric annotations, but we can change that, such as here, we want batch to be a group. We can also delete, add, or modify any annotations at this stage. Saving the changes returns us to the study. Now let's explore the default UMAP. Select the UMAP and it appears in the lower right. Use the Maximize View icon to see the UMAP in full screen. We can change the look, including the point size for each cell and hiding the legend. By default, our cells are colored by sample. We can also color by the expression of any gene in the dataset, such as this mitochondrial gene. We now return to the standard view to run more analyses. From the raw data in dark blue, we can run custom normalization. By default, the median normalization was used, so let's use counts per 10,000 instead. In the same dialog, we can also run PCA and UMAP or TSNE on the new normalized data. Here we input the same parameters as the original publication. The TSNE can then be viewed and modified the same as the default UMAP. To explore batch effects, we can view individual samples on the TSNE. Note here how the cells somewhat cluster by sample. This may indicate batch effects, and an integration was applied in the original study. We can perform integration to remove batch effects from the normalized data in orange. Europa has Harmony, BBK Nearest Neighbor, Combat, and Scanorama integrations available. Notice the Only Use Selected Samples option. Here all samples are selected, so all are used. With the new integrated dataset, PCA and UMAP are also run by default. If you want to run each step individually, any normalized dataset in orange can be used to run PCA or UMAP and TSNE. Similarly, any PCA in blue can be used to run UMAP or TISNE. Here we'll run TISNE from the integrated data, similar to the publication. Using any UMAP or TISNE, you can then perform clustering. Europa utilizes the Leiden method and allows you to do up to 6 unique clustering resolutions at a time. We explore the clusters in the TISNE. Select clusters by clicking in the plot or the legend. Change clustering resolution in the drop-down menu. Next, we'll annotate the clusters to cell types. For automatic cluster annotation, Europa supports CellTypist or Pangolao databases. Here, let's use a COVID PBMC dataset since these data are all PBMCs from COVID patients. You can learn more about these reference databases from CellTypist. Selecting the annotations, once complete, our TISNE is now labeled. Here I'm selecting all the monocyte clusters. Notice how the teal CD14 monocyte annotation encompasses multiple clusters. We can further resolve cluster annotations by hand. Again, we're going to explore the monocyte clusters. Let's select all of them in the TISNE and give them temporary names similar to the CellTypist ones. These names are not saved until we run the final by hand annotation, so we can modify them and update them until we're happy with them. Next, we can use the marker gene plot to determine genes that help refine our annotations. We can visualize these markers or selected genes of interest in the TISNE. Here, we view CD14 as a classical monocyte marker and see that it is more highly expressed in the teal 7 cluster. Thus, we update our cluster annotation. This process, in conjunction with automatic annotation, can then be repeated for all clusters as well as at different clustering resolutions as desired. This process is one of the most time-consuming in single-cell analyses, but with Europa Data Explorer, you can easily combine automatic and by-hand annotations to robustly define your cell types, as well as modify them over time as you learn more about your data. Now let's consider statistical analyses of this dataset. Remember that we already added sample annotations for this purpose. In the columns quadrant, group by one or more annotations of interest, then add a column set for analysis. Select that set and select statistical test icon. First, we select 2 groups to compare. Then we select the normalization method. When 2 groups are selected, Wilcoxon, t-tests, and regression tests are available, as well as multiple comparison corrections. Finally, we select the clustering resolution and minimum cells per group for analysis. If we instead select 3 or more groups, the all-by-all methods are available. These will compare each pair of groups. You may be wondering why normalization is a parameter, even though we've already normalized and integrated these data. This is because statistical tests are run from unintegrated counts. This is standard for single cell, as integration is aimed at modifying the UMAP or TISNE, not the counts. Let's go to the original publication for a second. The authors identified a natural killer cell population, in teal here, that expressed CCL3 more highly in COVID-19 patients. We can explore the same result in Europa. First, we find our natural killer cell cluster 2. Then we view the marker gene result for CCL3 in cluster 2 from our statistical analysis. Though expression is rare, CCL3 is more highly expressed in cluster 2 for COVID-19 patients compared to controls or flu patients. We can return to the t-SNE to visualize CCL3 expression at the cell level as well. Remember that our clusters are not identical to the publication, and more refinement would be required for an exact comparison to the publication. Taking a more global view of the statistical test results, we can view the volcano plot. Because we ran an all-by-all analysis, we can look at each pairwise comparison. We use the drop-down menus to change the comparison. Using slider selection, we can then define differentially expressed genes, or DIGs, by fold change and significance. Since we're on a negative log 10 scale, a value of 1.3 is roughly p less than 0.05. We group all the upregulated genes above these cutoffs into a row set. We'll define it as a gene set specifically so that we can run pathway analyses. Row sets can then be viewed in the lower left quadrant. We select a row set and then run Fisher's pathway to compare our DIGs to known pathways. All of the Broad molecular signatures databases are available for human and mouse. Once complete, we visualize the results under the biological context section. There are several visualizations, including a dot plot, showing the gene ratio, or K over K, and significance, as well as the results table listing the genes of interest in the pathways and all of the results. And there we have it, a complete single-cell RNA-Seq analysis including QC, normalization, clustering, annotation, DIGs, and pathways. We hope you enjoy analyzing your data in the Europa Data Explorer soon.